Good afternoon, everybody. My name's Tom. I'd like to welcome you to Nature Live. And for those of you who've never been to a Nature Live before, we're a daily event here in the Attenborough studio, and we're a chance for visitors to the museum to find out about some of the science that goes on here, because as well as being a wonderful place to visit and a great place to enjoy, the Natural History Museum is also a centre for scientific research and collections. So behind all of the walls and the closed doors of the galleries that hopefully you've been enjoying today, we have a team of scientists, and they work with a collection of millions of specimens so that we all understand a little bit more about this amazing planet on which we live. And today we're very fortunate to be joined by one of our scientists. We have Beulah Garner from the Department of Life Sciences, an entomologist in the Department of Life Sciences, I should say. And she's going to be telling us a little bit about a recent trip she made to Peru and a little bit more about the beetle collection which she looks after. We're very informal in Nature Live, so any questions you want to ask, anything you've ever wanted to ask about beetles, you might not have thought of anything before coming here today, but you've got a minute now to have a think. Pop up your hand and I'll run over to you with the microphone so that everyone can hear. We've got about 20, 25 minutes. Well, thank you firstly for joining us, Beulah. Could you start off by telling us what you do here at the museum?
Yeah, that's always the most difficult question to ask, actually, because we do so much. I am one of 5 curators that look after the beetle collection here in the Natural History Museum, and our collection has about 9 million specimens. So I've got responsibility for about 20% of that, so you can imagine that that's more than my lifetime. I'm sure there will be specimens in the collection that I will never see because there are just so many, and we're collecting them all the time, as you will learn today. What else? So we have about 250,000 species of the world's known 400,000 species. So, you know, we've still got a huge job to do. So it's really important that as a repository of biodiversity, we continue to collect and name species when we find them in exotic places around the world.
And you said earlier that you look after this collection. What does that mean when you look after it?
Well, we have a sort of duty of care because we're, I suppose we're funded by the government, so we belong to you, really. You know, we look after the world's species for you on your behalf and the world's behalf. So we have to make sure that they're stored in the correct environments, and most importantly, we make our specimens accessible to researchers all over the world. So in any one year, the Department of Entomology has about 300 international researchers that come through our doors and use our specimens. And the specimens that we have are very important because we care for them where other countries may not be able to. They may not, say in developing countries like Peru, in some places they may not have the resources or the finances or the institutions even to look after their biodiversity. So we act as a repository for that.
And you mentioned you look after part of the beetle collection, so I think first we should have a scientific definition of what a beetle is. You brought a drawer that shows how different they can look, but what is a beetle?
That shouldn't be a difficult question. A beetle is an insect, so it has 6 legs. It's divided up into 3 body parts, so there will be a head, a thorax, and an abdomen. Their main characteristic, and where the name Coleoptera comes from, is from their wings. So what they have is a hard outer pair of wings, which are known as elytra, and underneath that is a veined pair of wings with which they fly. Most beetles do fly, many don't. And the name Coleoptera comes from the Greek, meaning coleo, meaning sheath, and ptera, of course, meaning wings, so they're sheathed winged. And they also have biting mouth parts as well, so many of them will be predatory, many of them will feed on flowers or leaves or dead wood, whatever. So that's about it, really.
And within the beetles, a particular specialism of yours is the scarabs. And so could we find out exactly what they are? We've got another drawer to show them.
Yep. Yeah. Can we see that? This is a sort of random display drawer of lovely, lovely beetles that are not Peruvian, I have to admit. They're Tanzanian. A good colleague of mine collected them recently. But they show the diversity of the family Scarabaeidae. So here we've got Cetoniines, which are flower chafers, and here we've got the scarab beetles proper, which are dung feeders and dung dwellers as well. So that's just an example of the family Scarabeidae. There are 40,000 species that we know of in the world, so it's a huge, huge family. And yeah.
They are incredibly beautiful. And at the end, when you come down and have a look, you'll see these incredible colours. And it's not just us these days who think they're very beautiful. In history, the Egyptians, for example, they had a particular role for scarab beetles.
Yeah, the dung. Yeah, have we got some? The dung rollers. No, they're the special ones.
Okay, we'll look at these ones first.
You want to look at these ones? Yeah, oh, okay. They're what we call rutulines. So they're leaf. Can we see them? They're leaf chafers. So they mainly feed on leaves, and they're mainly found in tropical environments throughout the world. And please do come down and have a look at these afterwards, because they are proper Christmas beetles. They're our Christmas baubles, especially for you today. Some of them that we have are rare in that we've hardly been able to collect them over the years, and some of those specimens there are over 100 years old as well, so we'd like to find some more.
We're going to find out now about your most recent trip to collect beetles, but does anyone have any questions about beetles they want to ask at this stage? Yes, can I run up with the microphone super quick? Okay, there you go.
Hi there.
Hi.
What's the average life expectancy of a scarab beetle?
That's an interesting question. It varies between subfamilies, but with the scarabs, they are tend to be the longest lived in the larval form. So, like the grub, the sort of caterpillar-like grub, that's the part of the life cycle that feeds the most, and it needs to feed the most in order to metamorphosize into the adult. So, for example, our stag beetle in the UK, it can have a 7-year life cycle. The adult will only live for, say, a month, 2 months at the most, and the larvae can live for 5 or 6 years underground, dependent on food resources. So a lot of the time, their life cycle is dependent on the resources that they have in order to feed and complete the life cycle. But they can live for a year, or, as I say, about 7 years, it depends. In captivity, beetles will live for ages, for years and years and years.
You mentioned earlier that we're still adding to the collection that we have here at the museum. We're going to talk now about Peru. So while we fly over there with the map, could you tell us exactly where you were and why you went to this part of Peru?
Well, Peru for us is a very interesting country and locality, least of all because it has incredible tropical forests that are diminishing at an alarming rate. We really don't have that many specimens in the collection from Peru, and we've been collecting for 250 years, give or take a few years, and so it's really important that we highlight these places, these countries, where not much is known about the biodiversity of what they have there, particularly when precious habitats like rainforest are under threat. And if we think about 45% of the world's rainforests have diminished over the last, or have been lost over the last 100 years, which is really terrifying when you think about it. And that, of course, means that we'll lose species. Species habitats are much more reduced, and the whole ecosystem is under threat. So that's really why we went there, to go to a locality that we'd never been to before. I'd never been to before.
And we're going to talk a little bit about the process of collecting. Once you've identified where you want to go, how do you go about then targeting the species that you think you might be looking for?
Well, there are lots of different ways of collecting, especially with the scarabs. They're really good to collect because within their subfamilies, they exhibit very different lifestyles. So some, as I said, would feed on fruit, some on flowers or leaves or dung. So we tailor our collecting methods to suit what we want to collect. The best one to talk about is the dung, of course. All entomologists love to talk about poo, so indulge me.
Please, Beulah, be our guest. Explain to us how you use poo to collect beetles.
Yeah, can we have a
this
look at some?
This isn't Beulah going to the loo.
No,
it does look like that in small, but if you look carefully, it's
when
perfectly
Tom
fine.
first saw this picture, he said, "That's not you actually going to the loo, is it?" How rude. Of course not. But what I'm doing there is setting a dung trap. So some beetles are attracted to very specific dung. So you might get some scarabs that will only live or feed on elephant poo. Some are more general. But the trouble that we had in this environment is, it's a good example of it being under threat. It was suffering quite a bit from some tourism, local tourism, and also from cutting down the rainforest. So there was a lot of a cloud forest, I should say. There was a lot of sort of secondary forest, not much primary forest left. And so there are fewer mammals around. So we really struggled to find some dung to set our traps with. So we had to make our own. And I was on this expedition with 30 other people whom I'd never met before. And you start to get to know one another very well indeed, very quickly, when you have to discuss your toilet habits over breakfast. So, yeah, there we all are. And
you're being too polite and very, very ladylike about this, but basically you are using human poo just to collect the insects, and you form a trap, and that attracts the beetles, and they fall into it. That's the idea.
That's right.
And is that what's known as a pitfall trap, Beulah, or is that a different kind?
That's a different kind of trap, Tom.
We're not going to show you the ones collected with human poo, I'm hoping.
I don't think we are, no. Okay.
No. But we are going to talk about the ones that you get in pitfall traps.
Yes.
So can you tell us about how you set those up?
Yeah, well, they're similar to what you see there. I mean, that's a dung trap. That's a pair of my old tights that I very cleverly reinvented for the purpose. But a pitfall trap is a submerged cup with water and a little bit of fairy liquid, so that if any insects fall into the trap, they can break the surface tension, because some of them will be very light. And those are checked daily, because it's a cloud forest. You know, there may be foraging animals around, and maybe humans, and and also the rainfall as well. So we don't want our specimens to rot. So we would check them daily.
And any of the scarab beetles that you were looking for, do they ever fly? Do you catch them while they're in flight as well?
Yeah. There's an image of the fruit. Oh, this might be a particular method that we use. This is a fruit trap. Can I pass this?
Of course
round?
You can, yeah.
It's one I made earlier today. So what happens is the chafers are really attracted to sweet-smelling, rotting fruit. So they would just fly into the bottle, and this is a little rain shield here, and then they would fall in and hopefully have a very pleasant alcoholic death.
Please don't turn the bottle upside down, because it's rather smooth. But do have a sniff in one of the gaps.
So yeah, that's what they're attracted to, that lovely smell. So what you're smelling is bananas and alcohol. So you make do in the field. You devise very inventive methods of trapping.
We've got some other traps that you sent me photos of as well. I mean, this, for example, here, is this a different way of trapping?
Yeah, that is specifically for flying insects, flying scarabs. So what you would do is, you can just about see that there's a net there. So you would secure that across what you might think is a flight path of insects. So what you have to do is go into the forest and have a good look around first and see where you think is a good place that insects may be flying around. And so you set what we call yellow pan traps underneath the netting, and they're filled up with water. They're yellow because insects love yellow. They're attracted to it because they think it's a plant. And above that is a sort of canopy to keep off any rain or debris. And those also would be checked daily.
We've got a person in the second row. You seem to like the smell. We had a belly rub there after the smell. Did you like that smell there? That's okay. Watch out if you're a beetle, though, because that's not going to lure you to good things. Did you like the smell of it? Yeah? Yeah, I think so. You definitely... It looked like you did.
I'll give you something later that you won't like the smell of, I assure you.
Once you've collected the beetles, they're affectionately known in the trade as beetle soup. Is that
beetle
correct?
Soup,
And you've brought some. Could you show us that?
Yeah, so... once we've collected them in the field, we decant them into maybe a pot like this that has alcohol in it, very high-strength alcohol that, of course, none of us would ever drink in the field. And on here is written light, so this was taken from a light trap, which I think we've got an image of somewhere. But anyway, I'm just going to pour some of this out to show you some of the wonderful creatures that all of it out, wonderful creatures that we get in a trap.
And Beulah, how many insects like this might one hope to catch in a day, for example?
Oh, hundreds and hundreds. I mean, it depends. We time our trips for beetles to coincide with the rainy season because they're triggered. Their life cycle is triggered by the rain. So in the dry season and up to the beginning of the rainy season, they'll be in their pupal stage. So they'll just be in a little case waiting to emerge. And as soon as the rains come, they'll emerge. And that's hopefully where when we'll be there as well. So there's a huge diversity in this trap. And light trapping is particularly effective in the tropics because what you want to do is to be able to see what's happening in a whole 24 hours. So you're trapping all the time. In the daytime, you might be out with a net, sweeping things that are flying around. And then at the in the night, you would set up a light that would attract insects. Shall I pass that around?
We could pass that around, yeah, if it's happy to go. I mean, it's definitely incredible
as
to
long
see.
As
Don't spill it. I'm worried about our audience more than the specimens on this one because they'll smell terrible for weeks. I imagine it doesn't smell great.
This one doesn't smell as bad as the
Do you want me to pass that round?
Yeah.
And when you're going through your beetle soup in the evening, are you identifying all of the species, or is it a question of maybe not knowing exactly what you've collected that day?
Some of us would be better than that, more expert than others. On this particular trip, we were very fortunate. The people that organised it were the scarab experts in the world. So they came from America and South America, and they were really there to instruct us on how to identify, mainly to subfamily and below. So when you go out in the field, you've got an idea, one, of what exists in your collection, our collection here. And so if you see something different, then you can say, oh, I don't think we've got that in the collection, but you might not necessarily know that that's new to science. But if you're an expert in a particular group, then you could be out in the field and go, oh, I've never seen this before, which is quite possible because we're describing new species, like between 1,000 and 3,000 new species a year, which is an incredible rate, really, and really heartening. Yeah, there's so much more to discover. So,
especially for any young budding entomologists in the audience who think it might all be done. It's definitely not. There's plenty of work for you to be doing.
We definitely need more entomologists.
Does anyone have any questions they'd like to ask at this stage? I'll give you another chance. Yes, do you want to shout down, sir? Yeah, that's a really good point. Can you tell the difference between a female and a male beetle?
I've got an excellent example of that, actually. The scarabs are really good for that. They help us a lot. Have we got this on the visualizer? So these are what we call the rainbow scarabs. There we go. We might not be able to.
Do you want me to hold one up, perhaps, and to the camera?
Perfect. So these exhibit what we call sexual dimorphism, which is what you're talking about. So there's a difference between one of the big ones, the male and the female. So the males will have huge horns, and the females may have no horn or a reduced horn. And the males are really showy, somewhat typical. They use their horns to show off to the female, and they also use them to fight other males for female competition. So that for us is quite easy. There are other ways. We do dissections in the lab. Should I get out of the way?
Let's see if we can get this in. This one's an absolute beauty, so yeah.
Okay. We dissect them. We dissect their genitalia. So that's a really reliable, well, most of the time, reliable way of telling the difference between a male and a female.
And for those of you who were hoping that the tone was going to be raised, having talked about poo, we're going to take it right back down to the gutter and talk about genitalia. And we can actually show some beetle genitalia, and you'll see then, sir, the difference between beetles of different sexes. Have you got a good example of that? Maybe in this one here, Beulah?
Yeah.
Okay, if we pop that one on.
That's not the right lid. Where was it, Tom?
Well, we might have to come
One
back.
we saw earlier. This one.
Yeah. Okay. All right. So if I hold that
there
up,
and there.
if I hold that like that, we'll see if we can get it up on the camera.
This is a special preparation technique. It can be very, very fiddly. I mean, with the bigger species, it's not so difficult. But when you've got, some scarabs can be as big as 1 millimeter in length. So in order to dissect them, you have to have very high magnification. But if you...
Okay, so if I move it down a little bit, there we go.
Mounted on the end of that, that's it, that specimen, is what we call its adiagus. And that's a sort of scientific term for its Johnson.
There we go. So there you go, sir. That's how we tell the difference. Bet you wished you hadn't asked now. Any other questions that anyone would like to ask? Yes, can I come over with the microphone, sir?
When you go on an expedition, you went specifically to collect scarabs. Would you totally ignore any other specimens that are collected at the time?
What was the end bit? Sorry, I didn't
So
quite
would you
hear that.
ignore other species or families that
No,
you're...
not at all. No, we collect everything. Nothing is wasted. There's always going to be what we would call a bycatch, and so you would get other things other than beetles as well. And we bring all of that back, and we would distribute it to our colleagues. So if we collect some wasps, we would give those to the wasp curator, and the same for all other groups. And also the most important thing that we do is we return hopefully identified the specimens that we've taken from the countries where we've been collecting. So we have what we call, we have permits, and we have maybe a memorandum of understanding which would stipulate how much of the catch we give back to that the institution in the sort of main institution, our sort of sister institution, if you like, in any given country. So they receive back in very good order identified specimens as well.
And you mentioned that this is a very collaborative project, and there are lots of people from all over the world working on different projects who were with you at the time. So I suppose they would focus on different families and different kinds of insects.
Yeah.
When you're flying the specimens back to the UK or any other part of the world, you're not allowed to fly them in alcohol, are you? And you've brought back the way in which they're packaged, which is quite good and quite smelly, and it means you get a chance to have a look at them up close. So if we pass those round, it also coincides with this photo here, which is quite a good one that you sent, Beulah.
This is health and safety lab gear that we all have to wear when we're dealing with these horrible smelly specimens. This is my good colleague Lucia, who has mounted many, actually where is that drawer? Many of our specimens. Let's take some of these out. I'll pass this round. This is mounting in progress. I've digressed a bit, sorry. So this is how we pin.
But this is what they look like when they're being travelled around the world.
You will all have had your liquids confiscated, no doubt, when you go to the airport. Well, it's the same for us when we're transporting beetles. So we may have had them in a pot like this whilst we're in the field with plenty of alcohol, but what we have to do is take the alcohol out so there's only a minimum amount to keep the beetles, the specimens, safe and protected. And then we'll recant them when we get home. But these are rather old now, and you can handle them if you wish, but do wash your hands afterwards. And they do have a smell. They're horrible.
And often these beetles are sent to perhaps an unusual place to be mounted. There's a big conference that goes on in Prague, is that right?
Yeah, that is quite a spectacle. Twice a year, all of Europe's entomologists, including coleopterologists, gather together in Prague, and mainly to drink beer and eat pork, from as far as I can tell. But what they do is they exchange specimens and talk about their recent scientific work. And what we do, and here's an example of some wonderful fashion. The gentleman on the left always turns up in a pink suit, and the gentleman on the right, well, we won't say anything more about that. So we send our specimens out to Prague to be mounted because we're dealing with thousands and thousands of specimens every year. We simply don't have the time or the resources to mount them all, or pinned in the way that you will see with that box going round. So yeah, we head out every twice a year, and it takes about a year for these specimens to come back to us. So some mounters will identify what we send them as well, and that's always really exciting because we're always hoping for a new species.
And you mentioned finding new species. Is the work that you're doing helping to increase the known biodiversity of this area and in the end help it be preserved? Is it under threat, for example?
One would hope. It's part of what we do, really. I mean, taxonomy, which is the museum's main sort of currency, if you will, which is the naming and classifying of organisms, underpins all other sort of conservation sciences. It underpins ecology, it underpins conservation, because if you can't name something, then you cannot conserve it, and you can't use it as a sort of defense, if you like, for maintaining a habitat, a pristine habitat, or a habitat that is under threat or rare. And this was on the way to our site. This is a big scar on the landscape, a big mining activity going on there. And the trouble in many developing countries is that this sort of activity is unregulated, because they won't necessarily be aware of, or the government in power at the time may not even care about their biological resources. So the work that we do can help other people in the wider sort of scientific community defend these habitats and conserve these habitats for the future.
We are running out of time. Yes, one last question. If I could run over to you with the microphone, you can take.
Conservational-wise, do you have any kind of keeping of genetical information about these animals, like any kind of genetic profiling of them?
Yes, we do. Yeah, we have a separate department in the museum. We actually have a new molecular collections facility. Collecting for molecular work is very different and difficult as well, because when you want to preserve DNA, you have to use a really high, like 100% alcohol, and you have to get the specimens back as quickly as you can. So there are methods that we could use in the field, like freezing specimens in liquid nitrogen, but you can imagine if you're in the middle of nowhere, it's going to be really hard to get the specimens back, and also get them back to the UK as well, because the flasks that we use look like they could be bombs. I don't know. We have trouble with that sort of thing, but we certainly do. I mean, we can even extract DNA from historical specimens. It is possible, and we actively do that as well. And increasingly now, it's important in science because it's helping to understand sort of evolutionary branchings of taxonomy, so we can have a more deeper understanding of what relationships species have in the world.
We have run out of time for today's event. If you have any extra questions for Beulah, or you'd like to come and have a closer look at the specimens, and I do urge you to come and have a look because they're absolutely stunning, do pop down because we're going to stick around for a few minutes. If you want to find out more about Beulah and our other entomologists' work, there's a fantastic beetle blog on our Nature Plus page. It's written in a very dry and hilariously funny manner, so you'll enjoy that. It's definitely one to keep up with. We've got quite a few contributors in the audience, and all of them are excellent orators when it comes to the beetle work. Before we go, however, I'd like to remind you that Nature Live is a daily event. We have a different speaker every day, so if you're ever back in South Ken and you're here at the museum and you'd like to meet another member of our staff and find out about a different area of work that goes on here, we'd love to see you again. Thank you very much for coming today. Thank you for your questions. And finally, and most importantly, please join me in saying a huge thank you to Beulah Garner. Thank you.
Thank you.